Great, so let's look at biological macromolecules. We're interested in them for a lot of different reasons. We're going to see how they really play out in biology, but one thing that is clear is that we find a lot of these macromolecules in the foods that we eat. So we're going to take a look at some of the ways that we can view them, these being the four major classes of macromolecules. So we're going to focus first on carbohydrates, but we're also going to talk about lipids and proteins and nucleic acids. All of them are organic compounds, therefore they all contain carbon. Remember, that's kind of the prerequisite for organic compounds. And many of them are going to also have carbon, hydrogen, oxygen, and some have nitrogen and some other elements. Here we can see some description of this, but what we're really interested in is the fact that when we look at a macromolecule, macro means large. So what we're looking at are much bigger molecules than what we've typically looked at. And we are also interested in this understanding of what is a monomer and what is a polymer. So monomers are individual subunits of some type of a macromolecule. And definitely carbohydrates, proteins, and nucleic acids all have very specific monomer units that then build these bigger units, these polymers. So the mer part means unit, mono means single, so a single unit, building poly, which means many units. Now lipids are a little different, so we'll get into those a little bit later and you'll see why. So monomers, single units, and they are going to be joined, and then we are going to look at how those units actually can also be formed into the bigger polymer units through this process called dehydration synthesis. So moving on, I think the picture tells us what we need to see. So here on the left is a glucose molecule. So this is a monomer. It's a monosaccharide. It's a unit of sugar. And glucose is then going to join here with its neighbor, which is also a glucose molecule, to form something called maltose, which is a disaccharide. So maltose is this thing that we see over here on the right, where they're joined together. But how they join is through the loss of a water molecule, and that's called dehydration, having to do with the water, synthesis, meaning putting together. So a dehydration synthesis is going to combine these two monomer units, monomer meaning in this case a monosaccharide, and another monosaccharide, and it's going to, through the removal of water, that we can see where that comes from, this OH on this molecule, this hydroxyl group here, and the hydrogen from the adjacent hydroxyl group are going to join and be removed, and the consequence of that is what you see over here. This is called a glycosidic bond. So glycosidic bond is what we'll talk about when we get more specifically into carbohydrates. But what it is, but the process is dehydration synthesis. And so you see this union of these two monomer units. Now we have a disaccharide here, and we have water that is one of the products at the end. Now, here's hydrolysis is the opposite. So let's take what we just talked about, maltose, and let's convert it back into those two individual units, those individual glucose units. And that also can happen when water is added to the reactant side of the equation. So you can see that water is on the reactant side. No longer is it on the product side because it's already now within these two individual monomer units. That is called a hydrolysis reaction. Sometimes it's also called a dehydration reaction. I like the term hydrolysis better because it doesn't get us confused with some of the other things we just dehydration synthesis. So dehydration synthesis is putting together, and hydrolysis is breaking apart, both involving water. Now, it can't happen at all just spontaneously. So there's something that is going to help this process at least to happen much more quickly than it could. And those things we're going to call enzymes. So we'll see some of that as we move forward. This is basically, again, showing the hydrolysis reaction. And so this disaccharide here on the left, which is maltose, is going to be broken into those two monomer units with the addition of water, okay, which is that hydrolysis. So water is going to split this back into its two parts. But it has to involve, in most cases, an enzyme. In this case, the enzyme would be called maltase. So when you see a word like maltose, OSE means a sugar. And if maltose is the sugar, and it's a disaccharide, it's going to need an enzyme that's very specific to that, that's going to help it to break down. And that's called maltase. Now you've seen this kind of thing already, or at least you're familiar with it, because you've heard of lactose intolerance, right? People who can't break down the lactose disaccharide in things like milk. And so for those people who have that, they may not have sufficient amount of the enzyme lactase, which is what's required to do that. We'll see this more when we get a little bit more depth into enzymes. So that's kind of the first section here, and I think we can move on a little bit here and we'll talk about carbohydrates. So, because in truth we've already looked at it, right? We've seen that carbohydrates are these things that we're familiar with, that a lot of us really love, and that they are composed of the elements carbon, hydrogen, and oxygen in this particular ratio that you see up here, this 1 to 2 to 1 ratio. So for every 1 carbon, we'll have twice as many hydrogen, and we'll have 1 oxygen. So that's the sort of the recipe for a carbohydrate. The smallest unit, the individual subunits of carbohydrates, are called monosaccharides. If you have 2 of them together, like we just saw with maltose, then you have a disaccharide. Di meaning 2. And then if you have bunches of them together, we call those polysaccharides. So there's lots of ways to classify sugars. We can just look first at the individual units, those monosaccharides, and we can look at them by how many carbons they have. So that's one thing we'll look at here coming up on the same slide. We can also use the suffix OSE on all of them to indicate it's a sugar. And we're going to see what's probably the most important way to classify them is this carbonyl group, or carbonyl group, depending on you want to pronounce it that way. But it's that carbon with a double bonded oxygen. Remember, that's a functional group we looked at. And there's 2 places that carbon with a double bonded oxygen can be placed in a molecule. And that's going to change what we call it. So the 2 names that come afterwards, this aldose and ketose, is referring to where that group is. And if it's at the very end of the chain of these carbons, what it's going to be called is an aldose, meaning it's an aldehyde, but because it's also, now we call it a sugar, right? So it's an aldose sugar, where the one that has the carbonyl groups in the middle of a carbon chain is under the category of ketones. Go back and look at your little chart on this if you don't remember that. And actually that's a good idea to just kind of see that. And so that ketone carbonyl group on a sugar will then make that sugar be under the classification of a ketose sugar. Remember, OSE means sugar. We can also classify it in terms of the numbers of carbons. So here, this is where that information you learned about earlier is helpful. If it has 3 carbons, it could be called a triose. If it has 5, it could be called a pentose. If it has 6, it could be called a hexose, etc. So all of these ways are classifying sugars. And what we're looking at here are the simple sugars, the monosaccharides. On the left, you can see here, where you see here is an aldose. On the right, you see a ketose. So notice the placement of that carbon with the double bonded oxygen up here, or in the middle for the ketose. If you look below, you're seeing those same, some of those same ones here, like here's the glyceraldehyde, which is that has 3 carbons. It's an aldose sugar. but it's also a threose, which means there's 3 carbons. The one next to it is a pentose, meaning 5 carbons. It also, though, is an aldose sugar. Look at it and see why. And then go to the right where glucose is in that box, and you see that it's a hexose, and it's also an aldose sugar, right? Because we're looking at aldoses on all cases there. They all have that carbon double bonded oxygen at the end of the chain. So all of these can be also considered, we can look at isomers. Remember this? So this is why we learned some of this in the earlier chapter. So structural isomers, if you remember, are just ones that have the same formula but some different arrangement of these in their molecule. So what I think you should do here is to look at these 3 pictures, if not here, at least in your book, and look at the differences. It's like the Where's Waldo thing again. Find where there is a difference between these molecules, between the glucose, the galactose, and the fructose molecules here. Now, there's more than one difference. They all have the same formula, though, right? But you may find a couple of other things that you might say are also differences, such as, are they all aldoses? Or is there any ketose being represented here? Now, how we see sugars very often in the body is in ring structures. So when you put a lot of these biological molecules in the body, particularly in sugars, you're going to see they are going to form in an aqueous solution. So that just means in watery solutions. They're going to form rings. 5 and 6 carbon monosaccharides is what we see often in the body. So they can flip back and forth. They can be a straight chain, like the aliphatic types, or they can form these more aromatic looking structures, these ring structures. And where they do that is kind of probably what you're not sure of. So I think I put a star on this one that didn't exist in your book, but to just show you that this carbon right here would have been the one at the very top of the straight chain if I was going to lay this one out again, and I'll show you in class. But it's at that point where this is going to cyclize, where it's going to form this ring structure is right here. And this is called something called an anomeric carbon. So where the ring forms, that carbon where that ring is going to be formed is called the anomeric carbon. And if you have an aldose sugar, in other words, that C double bonded oxygen is at the very end of that straight chain molecule, then this is what it will typically form, is this hexagon shape, the 6 carbon sugar, but notice it forms the hexagon. Where if you look below, what you're seeing here is a pentagon shape. And so the ones you see below, those would be the ones that would be in aldose form sugars. I'm sorry, ketose form sugars. So aldose above, ketose below, you get that pentagon shape. We're looking here at some other examples, but now they're giving us that term I was mentioning before, which is this idea that when 2 of these monosaccharides join by dehydration synthesis, releasing one water molecule and forming a covalent bond between them, that bond is called a glycosidic bond or a glycosidic linkage. Here's some common disaccharides. We'll look at these more in class. I'm going to try to keep this video as short as I can. So we'll look at some of them, and some of you already know about them. And then we get to polysaccharides, which basically are going to be different in that one thing, they have lots of them. They have lots of monomers joined together by glycosidic linkages, but the way in which they join can differ. And that's what we're going to focus on in a little bit. So you're going to see starch and what that is. Here's an example right here of something we'd call starch and how that can form. There's also a polysaccharide called cellulose, which makes up plant cell walls. And it will link a little differently. and on animal exoskeletons. So that's it.